Okay, this is not unlike some of the other ones we've seen. It's just a large multipolar neuron. The axon hillock is probably here. Nucleus nucleolus, the granular type appearance in the cytoplasm is Nissl. I can't, because it's not stained. Okay, what you see on this particular slide looks like a shadow of an unstained area, right, coming out of there. It may get blasted out in the projector is almost too bright. All right, I think you can see it on the ones in the laboratory, because this is like a copy of the slides. But you get just a little bit of a hint that there's some staining there. Barely. I mean, it's not staining like these. Okay, Raphael goes into the dendrites, but not into the axon. Now, you know, the dendrites are coming out of the plane of section here. You know, because those arborizations at the ends of the dendrites, so they're not going to be in the plane of section. These are just, again, silver-stained multipolar neurons, not a biggie to worry about. The silver stain is just picking up some of the cytoskeletal elements in the dendrites, and that's why you're seeing some of this fine, almost like a meshwork or fibrous type appearance on the slide.
What's that? Can you go back? What's that, like, on the bottom corner? What's that weird-looking cell?
This?
Yes.
I just think the way the plane of section is caught it, you're just getting a lot of silver stain in that neuron, that's all.
Oh, okay.
More of the same thing, just multipolar neurons and just the cell processes that are just staining this kind of a fibery type network. This is most likely a peripheral nerve, and they're cut in cross-section. So that's one, and that's another one, just cut in cross-section. Now, in these nerves, you can see nuclei that are staining, and these nuclei can be nuclei of either Schwann cells or fibrocytes. All right? Now, there's a bunch of connective tissue around these fascicles. That can be considered a nerve fascicle or a nerve bundle, as can that one. Now, I can't get, on this image, you can't get any further away to know what you've got, but it may be a safe bet to say that this is perineurium. This connective tissue is perineurium, okay, which is kind of going to be continuous with the connective tissue around the bundle of the axons, that whole bundle of fascicles. If you could get really far out away and you were, say, grabbing onto the nerve, like with a forceps or something, it'd be picking up the epineurium. Endoneurium could be around, would be around individual axons. Now, at this level of magnification, I can't really resolve endoneurium. Okay, all I can say is there are nuclei in these bundles, and some of the nuclei could be nuclei of fibroblasts, which are making the endoneurium. And some of them are nuclei of Schwann cells, which are myelinating the axons.
I thought that perineurium was the, I guess, the flattened nucleus surrounding.
Yeah, this is, to be sure, this is perineurium. To be sure.
But it also includes the connective tissue.
It could include this connective tissue. I don't know at this level of magnification what we really have there on the tissue. Okay, remember, it's not unlike muscle. The epineurium is surrounding the entire nerve that if you had a forceps or a probe, you could lift up. Okay, and that epineurium is going to kind of invest into the bundles and be the perineurium that's going to be surrounding the fascicles. the bundles of nerve fibers, but there are multiple axons in the fascicle. Okay?
That's not.
Yeah.
Isn't the difference is the perineurium is epithelium, right?
The perineurium is an epithelium type of cells, yes.
But the epineurium is connective tissue.
Yeah, that's not a major point.
Okay.
Okay, that's not a major point. All right, the cells, the nuclei of the cells that make them are going to be fibroblast type cells. We're not going to make a big deal with the epineurium, or the perineurium rather, is an epithelial type thing.
I was just wondering if that would give the two tissues a distinct appearance, such that you can tell them apart.
The cells that make up the perineurium, if they're truly epithelium, you could do a cytoskeletal stain for keratin, okay, and you could pick up that it's an epithelium-derived cell. All right, but that's not something that I'm going to ask you to worry about now. Okay, actually, if you can just think of it in the analogy to the muscle sheets, you know, epimysium, epineurium, perimysium, perineurium, endomysium, endoneurium, that's fine. Okay. This one is a silver stain. The dark staining things are actually probably white fat cells that are stained. The silver stain is being picked up by the fat. Okay. Once again, this could be part of perineurium. Can't resolve the individual axons in here to say what's endoneurium. Myelin picks up the silver, just like the fat picks up the silver. So anything that looks like it's black in here will probably be the myelin around the axons. And so you can't really resolve the endoneurium around those. Okay, and again, if we could make the assumption among friends that you're picking up the nerve bundle out here with a probe, okay, then you could legitimately say that maybe out here is epineurium. Okay, make sense? So we could maybe say the outside parts are epineurium, and it's kind of investing into becoming perineurium. Just a, again, nerve bundle in cross-section. Nothing that's a big deal. You remember in growths, oftentimes blood vessels and nerves run together, and that's all this is showing. Here's the nerve bundle, bunches of axons. Here's the blood vessel. Okay, the endothelium would be the flat layer of cells just on this layer of the blood vessel called the tunica intima. They're not responsible for this exam. Tunica media are layers of smooth muscle in the blood vessel, and then tunica adventitia, the outermost layer of the blood vessel, is connective tissue, and that connective tissue is continuous with the connective tissue that would surround the nerve. All right, there's a little bit of an artifactual separation of the structures there. And then the lumen of the blood vessel is filled with red blood cells, and you can even see nuclei of white blood cells. This is probably in an area like the cauda equina. So myelinated axons, here's the axon running down the middle. The blue around the axons is again connective tissue, would be connective tissue of the endoneurium. And these nuclei that you can see in various places can be nuclei, again, either of Schwann cells or the fibroblasts making the endoneurium. And the arrowhead there is pointing out the node of Ranvier. I don't like that slide. It's just an overstained slide with diluted blue. Okay, the arrowhead there is, again, also pointing out a node of Ranvier. I think the description makes note of a nucleus up there. And again, the nucleus is either a Schwann cell nucleus or a fibroblast nucleus. That's a dorsal root ganglion. Okay, I don't know where the spinal cord is. My sense would say that it's probably over here somewhere, with the dorsal root coming out and the ventral root coming out. Okay, and you could say that it's dorsal root because that's where the ganglion is. The ganglia for the ventral roots are not near the cord, right? They're further away from the cord. Okay, and just a higher power view of a ganglion. You can see neurons are pretty big cells. You can see a nucleus with a nucleolus, and you can kind of see the granular nature of the cytoplasm. This is in the cerebellum. White matter. And then all of this is gray matter. This is molecular layer. Of the gray matter, this is granular layer. Of the gray matter, and the large neuron cell bodies that you can see there are the Purkinje layer of the gray matter. Okay? And that's just a higher power view.